So let me start with the basic thing. What we have? We have something called hematopoietic stem cells. Hematopoietic stem cells. So understand, hematopoietic stem cells. These are the cells which are positive for a CD marker, that is CD34. These hematopoietic stem cells, like as you know, hematopoiesis, it will start like in from yolk sac initially. Then it goes to your reticuloendothelial system, which include a liver, spleen, lymph node, and ultimately at birth you will see the main site of hematopoiesis will become bone marrow. So these are actually pluripotent cells which can form different types of cells. So we can divide this hematopoietic stem cells broadly. I can divide this into two categories. Like these could be the cells of lymphoid lineage or myeloid lineage. So we call them as lymphoid stem cells, myeloid stem cells, or lymphoid lineage, myeloid lineage. Now when we have a lymphoid stem cells, so these are your lymphoblasts. So always remember, blast means immature. These lymphoblasts will convert into more mature form. That mature form is called lymphocyte. Simple. These lymphocytes are of two types. So what we have B lymphocyte and we have T lymphocyte. Simple. Now this B lymphocyte will further form plasma cells, and these plasma cells will produce immunoglobulin. So we can have immunoglobulin, like initially we have IgM, that is the first immunoglobulin to be formed. Then this IgM will convert into different classes of immunoglobulins. That is a phenomena we call this as class switching or isotype switching. Means IgM will convert into IgG, IgA, IgD, sometimes it remain IgM and IgE. This immunoglobulin will convert into different class. First immunoglobulin that we have is IgM. Then this IgM will convert into different types of immunoglobulin, like it can convert into IgG, IgA, sometimes it remain IgM, IgD and IgE. And this phenomena which I told in front of you, what we call this as class switching, class or isotype switching. So they ask this MCQ in microbiology, immunology, like which is the first immunoglobulin to be formed, that is your IgM. So this is about lymphoid lineage. So we have two cells like lymphocytes, B and T. These are formed by lymphoid stem cells. Now coming to myeloid stem cells, these myeloid stem cells will have different precursors. These myeloid stem cells, this will have different precursors, or I can say different cells which are originated from this. So this can give rise to megakaryoblast. This can form second type of cells. We call them as monoblast. Then we can have myeloblast, and what we have erythroblast. Okay. Okay, now understand this erythroblast. These erythroblast cells, they will further form erythrocyte. So erythroblast, first they will form reticulocytes. So there are several phases. So that you have studied in pathology. Like there are early normoblast, pre-normoblast, and what we have here late normoblast, reticulocytes. So this will form reticulocytes. Reticulocyte is important, and this will ultimately form erythrocyte. So what I can comment here is like a reticulocyte, these are nothing, these are baby form of RBCs. So if you ask me what is reticulocyte, so remember reticulocyte is nothing, these are baby RBC. So in simple language I can tell you reticulocytes are nothing, these are baby RBCs. This myeloblast will further proliferate to produce National Board of Examination. Not a joke, it is NB means neutrophils, basophils, eosinophils. So neutrophils, basophils, eosinophils. Monoblast will form monocyte. So now you can see our entire WBC is complete. WBC means what we have in WBC: basophils, eosinophils, monocytes, lymphocytes, neutrophils. Maximum percentage what we have neutrophils followed by lymphocytes. Basophils, these are the least in number. Then we also formed RBCs, this erythrocyte, these are called RBCs. So WBC is formed, RBC is formed. Now this megakaryoblast, this will produce megakaryocyte. Megakaryocyte, the extension of cytoplasm of this megakaryocyte, this will produce a cell that is called thrombocyte or platelet. Thrombocyte, or we call this as platelet. Okay, why I am teaching it is very important in the initial slide. First MCQ is hematopoietic stem cell. These are positive for which CD marker CD34. Okay, then if I talk about hematology, what are the aspects that we need to touch here? In hematology, we have to discuss RBC disorders. The disorders of this component RBC, what we call them? We call them as anemias. If you see disorders which involve, like suppose in many infection, you will see this myeloblast lineage will be affected. Like eosinophil counts are high in many parasite infection, in allergies, in asthma. Then this monocyte will further form macrophage. So macrophage are produced from monocytes. Then in the same like once we under anemia part, then I'll teach you platelet disorder, that is thrombocytopenia, thrombocytosis. These are disorder of platelet. Clear? Then what we have to study? We have to study about lymphocyte problems. So the lymphocyte problem is, it could be a B lymphocyte problem or T lymphocyte problem. These are leukemias and lymphomas. So leukemias and lymphomas, these are basically problem of your lymphoblast. Okay, then one more thing, like here we will study. So this is like anemia part 1. This will be like infection part. So that is we are discussing in infectious medicine. Then you can see this together. WBC, this will form WBC. So this is like WBC problem, what we call leukemias and lymphomas. Third problem will be platelet disorders. And fourth problem you can notice here, these are the small cells. I am highlighting them now. That's a fourth condition here. The disorders of plasma cells. These disorders of plasma cells, what we call them plasmacytoma, multiple myeloma. Is it clear? So that is why you can see more plasma cells means more IgM, more IgM means more class switching, more class switching means more immunoglobulins. So that's why what we see M spike, right? We see M spike in IgG area. This is like, we'll discuss entire thing. So, but the basic information is very, very important. So if you ask me like what is this baby RBC? Baby RBCs mean these are I can say kiddish form or childish form of RBCs. That is called reticulocyte. Similarly, what we have a baby platelet. So I am writing this as baby platelet. So baby form of platelet is megakaryocyte. So there are disorders where RBCs are dying. Like suppose if RBCs are dying, this will give a signal to bone marrow. So bone marrow will produce more, will try to produce more. So baby form will increase. So that is why in hemolytic anemia, what we see, reticulocyte count is more. Similarly, in thrombocytopenia, what we see, megakaryocyte number is more. Bone marrow will try to produce more platelet. That is a struggle of bone marrow. So this is the introduction that we need to study. I want this to be clear, because now we'll move into the depth of hematology.